Hello everyone. Our next topic is linkage. So this is one of the exception to third law, that is third law of Mendel, exception to law 3, that is independent assortment. So linkage is an exception to third law, that is independent assortment. So what is linkage? So it is the tendency of genes to stay together during inheritance. So usually what happens in inheritance, when parents are crossing over, so when the gametes are crossing over, so the genes will separate. That means the genes has to assort independently. So what happens is in linkage, for example, if I am taking a dihybrid cross, two characters each, so they have to separate independently. But here what is happening? The genes will stay together during inheritance. That means there is no crossing over taking place. So throughout the generations they stay together without any separation. They do not separate. And why they are not separating? Because they are being present on the same chromosome. Like for example, if I take any two chromosomes, chromosome 1 and chromosome 2, so this chromosome, for example, round yellow character, and the other chromosome wrinkled green character. So usually what happens is, so there is a crossing over. So independently as like that. So what we will get is, we will get capital R capital Y, capital R small y, small r capital Y, okay, and small r small y. So these are parental combinations. That's what we can see in this. Parent, one parent is having capital R capital Y, so that is parental, and this another parent small r small y, that is wrinkled green. So parental combinations are there, and recombinations because of chiasmata formation. So meiosis 1 we have this in meiosis. So chiasmata formation, crossing over of genetic material takes place. So what happens is in linkage, sometimes because the chromosomes are present, because the genes are present on the same chromosome, sometimes the genes are present on the same chromosome, that's why they are not separating out. These two are not separating. If they separate, then only crossing over takes place, recombination takes place. But if they are not separating, so they will not allow for recombination. That means they both will stay together only, and this both will stay together. So there won't be any crossing over. So this is called as linkage. So because of linkage, what happens is there won't be independent assortment, and there won't be a dihybrid ratio. So dihybrid ratio we have 9:3:3:1, that is this 9:3:3:1. So there won't be any dihybrid ratio as such. So there will be again there will be only monohybrid ratio, that is 3:1 only. So no dihybrid ratio, only 3:1, that is monohybrid. So this is all about linkage. So linkage is of two types, okay, complete linkage and incomplete linkage. So we will be doing this now. In complete linkage, what happens? Complete linkage. Okay, in complete linkage, what we can say is the genes are located in the same chromosome. So since we are doing linkage, so this definition follows. The genes are located in the same chromosome and do not separate and are inherited together. Why? Because there is no crossing over, absence of crossing over, because of absence of crossing over. So in complete linkage, so the genes will combine together, so there is no crossing over at all. So what happens is, like, I'll draw a diagram and show you. But this is what complete linkage looks like. I'm taking parents. One is dominant in both the characters, another parent is recessive in both the characters. So what happens after meiosis 2? So there will be replication and the complete monovalent becomes divalent chromosome. Okay, and then see here you can see there is no crossing over taking place between the parents. So what happens is the chromosomes are inherited as they are. So one chromosome is carrying dominant, another chromosome is carrying recessive, and then these divalent chromosomes are separating as monovalent. So we are getting 4 daughter cells or 4 daughter chromosomes. Each chromosome, if you see again, 2 of the chromosomes, when the divalent chromosome is separated, so 2 chromosomes are dominant and 2 chromosomes are recessive. So here only parental combinations are observed. In complete linkage, only parental combinations are observed. Only parental combinations are observed. So there is no crossing over. So at this place there should be crossing over, but there is no crossing over. So if crossing over is there, then there will be another combination, that is capital A with small b, small a with capital B. There will be a crossing over. So here there is no crossing over. That's why only parental combination. There is no recombination. So if the genes are present on the same chromosome, if 2 different genes are located on the same chromosome, so they do not separate. If they are separating, then only recombination takes place. So in complete linkage, what happens is their genes are not separating, therefore there is no crossing over. Then what will happen? The parental genes are only carried out to the next generations. So next generation again only dominant, only recessive. So this is what is observed in complete linkage.